All right, class. So I want to clarify one thing before we move forward. At positive 30, the voltage gated sodium channels are inactivated. So I should erase this. Why cannot I pick erase? Anyway, so that's not true. At positive 30, the channel is, or the gate is inactivated. That's what happens at positive 30. Okay. Okay. All right. So this state is positive 30. And then this is closed, where the channel is closed, as you can see. So that's closed. And this is open. And this is inactive. Okay. So this slide is just a review slide, and we went over all this information. Moving on to voltage gated potassium channels. Again, we have talked about this. So let's look at the PowerPoint and go through the PowerPoint real quick. Voltage gated potassium channels, they open at around positive 30 millivolts. And as they open, potassium moves out of the cell. We lose positive charge, bringing things back to resting membrane potential. And the process is repolarization. Then we have voltage gated calcium channels at the axon terminal, and hopefully you remember this from the muscle lecture, right? So as we have the action potential reaching the axon terminal, we have these voltage gated calcium channels that open up. Then calcium enters the cell. Calcium causes the synaptic vesicles to fuse with the presynaptic membrane and exocytose acetylcholine into the synaptic cleft. Acetylcholine binds to the ligand-gated channels on the postsynaptic cell and causes entry of sodium. Do you remember that? Okay, so those are the voltage-gated calcium channels that open up in response to an action potential. Quick review of the anatomy of the neuron. You have learned about this in anatomy, and I expect you to remember that information. So I will go over this super quick. Looking at the neuron, we do see, we were moving from top to bottom, we do see some cytoplasmic extensions, and the cytoplasmic extensions are called dendrites. I want you to know that dendrites are the receiving ends. Information travel towards the dendrites as shown by the red arrows here. So dendrites receive and bring the information towards the cell body. The bulb-like structure is the cell body, or also known as soma. And then if you look, the long structure that comes out of the cell body and extends, that is called the axon. The axon then branches to form these channels, or not channels, these branches are called telodendria, and each telodendria terminate in these knob-like structures, and the knob-like structures are called synaptic knobs. There is another very important structure that I want my students to know. Let's look at the junction of the cell body and the axon. So this junction is called axon hillock. In anatomy, we just learn axon hillock, but when we are talking in regards to physio, know that the axon hillock is also known as the trigger zone. Trigger zone, same as axon hillock. So I will change these two. I will use these two terms in an interchangeable fashion. And the axon hillock has actually great significance. It is highly important because it is at the axon hillock that the action potential is generated. Remember, at the axon hillock, we have the action potential generated. And the action potential, once generated in the axon hillock, travels all the down to the axon, all the way down the axon, and then reaches the axon terminal and the synaptic knobs. Alright, in this case, we also see that the axon is myelinated. The light blue tapes that are wrapped around the axons, those are called myelin sheath. And in between pieces of myelin sheath, right here, do you see? In between spaces in yellow, these are called node of RAN-VA, not labeled here, I'll do it for you. Oh, it's called neurofibril node, but we will just call them node of, RAN-VA. Okay. Now, neurons, do they talk to each other? Do they communicate with each other? Yes, they do. So, okay. They do carry electrical signals, right? They carry and transfer the action potential. Now, the action potential is generated in the neuron, or specifically in the axon, by opening and closing of many gates, right? And I like to call it dancing of the gates. We will see how the gates dance to open and close, and that leads to influx and efflux of different types of ions across a membrane. But before that, I want my students to know the basic types of channels that we find all over the neuron. And the types of channels are, we have sodium-potassium pump. Where are they located? They are located everywhere. Dendrite, soma, axon, hillock, axon. They are present everywhere. Then we have voltage-gated sodium channels. Then we have voltage-gated potassium channels. Then we have ligand-gated sodium channels. And finally, we have leak channels. We have sodium leak channels and we have potassium leak channels. Rupa, that's too much, I know. Okay, so we know that we have sodium-potassium pumps everywhere. We also have the leak channels everywhere. Okay. Now, if you think of the dendrites and the soma, so all over this place, dendrites and soma, sorry, I drew it wrong, very wrong. Here. Okay. Here. So all this part have ligand-gated sodium channels. Okay. All right. Axon hillock will have voltage-gated sodium and potassium channels. And the axon will also have voltage-gated sodium and potassium channels. So starting from the axon hillock all the way down, we will have voltage-gated sodium and potassium channels. Remember, we have sodium-potassium pump and the leak channels everywhere. And then looking at the axon terminals, we will have voltage-gated sodium channels, voltage-gated potassium channels, and also voltage-gated calcium channels. Those are present in the axon terminal. So if I have to draw an axon, modern art. So on the soma and dendrites, we will have ligand-gated sodium channels. At the axon hillock, and all the way down, all here, we will have voltage-gated sodium and voltage-gated potassium channels. The axon terminals will have voltage-gated sodium, voltage-gated potassium, and voltage-gated calcium channels. And remember, sodium-potassium pump and the sodium and potassium leak channels will be everywhere. Dendrite, soma, axon terminal, everywhere. Okay, let's move on. So, quick review. So, looking at the dendrites, know that they're the receiving end of the neuron, and we will have ligand-gated channels on the dendrites as well as on the soma, along with sodium-potassium pumps and the leak channels. Looking at the cell body, it contains a nucleus and the nucleolus as shown in the picture. Within the soma or the cell body, the neurotransmitters are produced, and the neurotransmitters then travel all the way down the axon and they remain encapsulated within synaptic vesicles at the axon terminal. And the neurotransmitter that actually bind to the receptors on the cell body is acetylcholine. Remember, acetylcholine works as a ligand that opens up the gate. So obviously on the cell body we find ligand-gated channels, and acetylcholine is a ligand that binds to open up the channels, and as those channels open up, we have sodium entering the cell. Well, and also soma has sodium-potassium pump and the leak channels. Axon, looking at the axon, they are nothing but the extension of the soma, and they carry the action potential down to the downstream cell. Axon axon has voltage-gated sodium and potassium channels on top of the sodium-potassium pump and the leak channels. Now, here we are looking at two types of axons, right? One has myelin sheath, the other one doesn't. Now remember, myelin sheath helps to insulate the axon, meaning if an axon has myelin sheath, the axon is obviously better insulated, and the charge doesn't dissipate, right? There is no dissipation of charge, meaning the charge doesn't just move outward. And because of that, an action potential travels way faster in a myelinated axon as compared to a non-myelinated one. In a non-myelinated axon, as you see in the bottom, which does not have the green myelin sheath, conduction of action potential is much slower, and of course most of the charge dissipates to the extracellular space. Okay, so looking at the axon terminal, those are the bulb-like distal ends of the axon. We have voltage-gated sodium, potassium, and calcium channels here. And then we also have sodium, potassium pump, the leak channels here. And this is a site of neurotransmitter release. This is where the neurotransmitter acetylcholine or other neurotransmitters are released. So the axon hillock, as we mentioned, it's also known as the trigger zone, and it's a transition site between the axon and the soma, and things work as all or none principle. So let's talk about that a little bit. Well, I'll talk about that in my next